I do want to thank the organizing committee members for allowing me to speak today, and we'll start. So as you know, era of big data and machine learning has arrived. And right before we speak, let's just kind of go over the terminology that I'll be using. So machine learning is a subset of artificial intelligence, and deep learning is a subset of machine learning. And data science what really encompasses all these domains. So a lot of what powers this AI revolution right now is deep learning, because these concepts of machine learning and AI have really stayed with us or have been with us for the past few decades. So before the era of deep learning, it was very difficult to write a program, say, to recognize this car, to recognize some pictures or analyze text. It would require very carefully constructed filters that would only work on a small set of images or under very restricted settings. But deep learning is very different. Deep learning learns directly from the raw data itself and changes its parameters to give the correct output after training. So it works best directly on raw data. So what is this hype about AI/deep learning? It's because the era of deep learning started really around 2013 with AlexNet, but didn't start until this hype until it started beating human performance around 2015. So as deep learning, as a technology matured, the number of medical papers using deep learning really exponentially grew at the same time, starting in 2015, and you can see how much it grew. So this has led to what many people call this fourth industrial revolution. So we have this convergence of computer hardware capable of storing and processing huge data sets, and the methods to analyze them now with machine learning and deep learning. This is all really exciting, but we do have to think about this curse of dimensionality. So one thing that is worth emphasizing here is that when people say big data or a lot of data, that can usually mean two things. Having a lot of information about a few number of people, which we usually call bad big data, versus having a lot of people with relatively fewer measurements or variables. For most machine learning, having a lot of independent samples is much better rather than just having lots of data on few number of people. So what type of big data are out there? I think UK Biobank is a tremendous example of a huge successful big data. It began in 2006, and it has recruited over 500,000 people from UK. So this has generated over 15 petabytes of data, over 2,000 global research collaborations, and it has already published more than 1,400 papers, and 200 of them have been in either Nature journals or Nature journal families. This is an example of a paper that has utilized eye data from UK Biobank. So they were able to just look at the fundus photographs of the retina of people and generate models that were able to predict cardio risk factors that we didn't think that they were not possible just based on retina photos. That meant age, gender, BMI, hemoglobin A1C, and then also even the likelihood of developing heart attacks and other cardiovascular outcomes. One of the things that we always think about as data scientists is the concept of bias, and how do we account for that? Let's take UK Biobank as an example. It was a convenient sampling, so all those 500,000 people were volunteers who were willing to come in and get all this data taken. Therefore, even common diseases that you would anticipate, they were very rare in UK Biobank. And they were heavily white British. So I think this figure will hone the big concept or this issue of bias. When they looked at the all-cause mortality rate, of people who are in UK population versus people who are participants in UK Biobank, there were up to 30% reduction in overall mortality rate. So think about what would happen if we were just using these data sets to train your algorithms, and then really, and then we have to think about how generalizable and applicable would that be in your patient population. And one more big limitation in our mind is the access, or getting access to that data set is definitely non-trivial. You have to know exactly what variables you want at the beginning, because if you want to add one more variable, you will kind of get started again in this process. You have to know exactly who your collaborators are, because you cannot add one more collaborator or site. And you have to get MTA and all that, and usually takes 1 or 2 years to get that data set. So it's really exciting, the NIH mandate of making all data fair, findable, accessible, interoperable, and reusable. And I think this will definitely expedite our data science research. So NIH has recognized this potential of using machine learning in healthcare research, but recognize all these limitations that exist in current existing data sets. So we mentioned this skew data. So for example, if you are just using EMR data, then they are heavily skewed towards disease participants or patients, obviously because they come to hospital because they have issues. If you rely on registry data, then obviously you're very heavily skewed towards healthy data, and that would cause bias. And also access is an issue. So NIH created this program called Bridge2AI to generate AI machine learning ready data set that is meant to be hypothesis agnostic, but then really immediately useful and shareable for future AI research to happen. And we'll be developing tools and standards and generating next generation of AI researchers. And that was what they required. And we're very fortunate that we are one of 4 funded programs in the nation. Our program is called AI Ready and Equitable Atlas for Diabetes Insights, or AI Ready. So I'll talk briefly more about our project. So the goal of this project is to create a multidimensional, ethically sourced data set in diverse people for studying salutogenesis in type 2 diabetes. In healthcare research, we are very familiar of studying pathogenesis. So we are very used to asking, what are the risk factors? What are the environmental, genetic, and behavioral risk factors that push people from healthy stage to disease stage? Salutogenesis is the reverse process. What are some factors that we can study that promote people from disease back to health? This is a huge project. So Aaron Lee is the PI, and I have the privilege of serving as a PD. There are 6 modules in this project. Teaming module led by Stanford team. Ethics module being led by Johns Hopkins University and University of California, San Diego. And they will be really discussing ethical issues of AI research. Standards being led by Johns Hopkins University and UW as well. Tools module being led by California Medical Innovation Institute, or CALMI, and OHSU. Dr. McWhinney, who will be moderating a session later today, is one of the PIs of that module. Data module being led by University of Alabama in Birmingham, and skills and workforce being led by UCSD as well. So to study this salutogenesis for our project, we are borrowing this previous success of single-cell RNA technique, where powerful machine learning models were able to do dimension reduction to place cells that are very similar to each other next to each other, and to place cells that are different from each other far from each other. And because we are able to, because we have such a big range of cells undergoing transformation, in the pseudotime manifolds, we are able to actually visualize how cells are moving from embryonic stage to a totally differentiated stage. So we are hoping to take a similar approach of cross-sectional sampling of 4,000 participants across even number or even stage or even spectrum of diabetes severity. And by approaching this as pseudotime manifolds analysis, we'll be able to discover many pathways that people move from healthy to diseased or diseased to healthy, thereby creating this atlas to study either pathogenesis or salutogenesis. So the project involves in type 2 diabetes, but really this concept or our methods can be applied to studying any other diseases, such as cancer. We are really addressing the limitations that I've mentioned, such as being unbalanced data set. So we're balancing in race and ethnicity in 1:1:1:1 ratio of white, black, Asian, and Hispanic. We're also balancing the sample in normal, no diabetes versus diet-controlled or borderline diabetes versus medication-controlled versus insulin-dependent. And we are also binning them equally in men and women. Data collection sites will be University of Washington, UAB, and UCSD. And also we are participating in discussions with American Indian population, especially the Cheyenne Riverside tribe, so that we can create foundation in getting participation from American Indians. So what are we collecting? So I just mentioned this huge data set. So we'll collect medical history along with previous electronic medical data sets, medical data that exists from participants. We'll do demographics and vitals, blood and urine for any biomarkers of diabetes-related outcomes. Lots of surveys, social determinants of health, cognition, NEKG, visual function, visual acuity, contrast sensitivity, lots of eye imaging because we are ophthalmology biased. And then also we'll do activity and heart rate monitoring, continuous glucose monitoring that will check your blood glucose level every 15 minutes, environmental sensor that they will take home and we'll be able to measure pollution data, any light data, and lots of sensor. And also we'll do whole genome sequencing on everyone and bank the samples for plasma, serum, and cryogenic storage of iPSC cells as well. And we're going to be open sourcing all the data. So the goal is that we generate this multimodal data set on this balanced sample. And the idea is that a lot of AI researchers will be able to leverage this data set to study cross-sectional sample for developing hypothesis generating and then also discovering some biomarkers to see what are the predictors or what are the biomarkers of diabetes severity or cognitive function, or those are some examples. And using the pseudotime manifolds approach, we're hoping that AI researchers will be able to leverage this data set to do predictive AI models. So they'll be able to understand what are some drivers of salutogenesis. And additional goals are that I mentioned that there's an ongoing engagement of American Indians, and we're hoping that they will be able to participate in this. And then also any new data that get generated because people are leveraging this data, we're going to ask them to be reposited back to open source repository so that this will become better and bigger for everybody. And hopefully we're going to accelerate data science in medicine by serving as a flagship data set for studying salutogenesis and diabetes. I want to spend a few minutes by talking about our lab, Computational Ophthalmology. And this is the slide that I showed earlier today, and this is really what we do at Computational Ophthalmology: machine learning, data science, and big data. And this is what you would see in our website, so please feel free to go and check out. These are some of the examples of big data sets that we have used so far in our research. I do want to highlight the IRIS registry or Intelligent Research Insight. We are one of the 5 academic institutions in the country that have research access to IRIS. This is a registry of all the medical EMR from ophthalmology or eye care providers in the nation. And we have records of 75 million unique participants and resulting in over 450 million patient encounters data. And it's thought that 1 in 4 Americans today, if you're seen by an eye doctor, then your records will be part of IRIS registry. So it's a huge data set. So some of the examples that we are able to do, we are able to study access to care or practice patterns, or study clinical outcomes of various diseases, or we are able to compare effectiveness of therapies, or this is really the strength of the big data, that we are able to discover new biomarkers or make new connections. We're using the eye as a source of other important critical diseases, systemic diseases such as Alzheimer's disease or diabetes. And we are able to apply some novel deep learning applications. And one of the exciting part in ophthalmology especially is this advances in imaging that are all non-invasive. And this is the work of Ricky Wang from Department of Ophthalmology and then Biomedical Engineering, where without any dye, we are able to visualize all this retinal vasculature and really allow us to characterize and quantify the level of retinal vascular health. This is the work of Ram Subishan from our department, optoretinogram, where he's able to stimulate one single photoreceptor cell and detect its response. So imagine how sensitive we can get to studying the health of the retina. This is the work of Yue Wu from our lab, in which he's able to instantly segment and quantify all the features, all the abnormalities in the retina related to diabetic retinopathy. And this is the work of Aaron Lee that's training the optical coherence tomography, this structural imaging to quantify the level of swelling that exists in the retina from diabetes. To summarize, this marriage of big data and machine learning allow powerful approaches to study health in ways that we didn't think that it was possible. We have just begun scratching the surface of this amazing potential, and we look forward to really talking with you and hopefully collaborating with many of you in the future so you can leverage our AI-ready data set. I do want to acknowledge amazing members of our lab who made all this work possible. Please wave your hand if you're part of Computational Ophthalmology so people know who to talk to. And I want to acknowledge our funding sources and also Sean Mooney and Tong Shen, who have been really grateful. And we're always looking for new talent, so please reach out to us. Thank you.